Hi class, welcome back to the lab. Today we are going to be going over microorganisms in the environment, which is exercise 2 in your lab manual. So what I'm basically going to do is just introduce you to where microorganisms are growing at, and then we're going to go over a little bit about media, which is how we grow the organisms in the lab, how we visualize them in the lab. So microorganisms are what we call ubiquitous. So that means that they're found everywhere. They're found in you, on you, in animals, on animals, plants, the ground, the water. Almost every place on the planet there is going to be a bacteria that is going to be using that environment to grow in. So bacteria are very, very important to us. They're important for our body. They're important for the environment. So it is important for us to learn about them. What are they doing? What kind of products are they producing? What kind of metabolism are they doing? What types of bacteria are growing in a certain community? Specifically, there's been lots of research done on the bacteria that are growing in our bodies and on our bodies that help our immune system and help our health. But the way that you study bacteria is different than the way that you would study a macromolecule, so in a macroorganism, which is one that's going to be large enough for us to see with the naked eye. A microorganism, which is a bacteria, is too small for us to see with our naked eye. So we have to take them and take a sample from the environment. We can take readings of it from the environment, but mostly you're going to have to come in, bring it into the lab, grow it up in the lab so that we can then do tests on it to find out what type of metabolism it's doing. We can do some molecular work on it to find out what kind of bacteria it is, or just classical micro where we're staining it and putting it under the microscope and looking at it under the microscope. But to do all those techniques, we have to usually bring it into the lab first and culture it and grow it in the lab. So to do that, we use what is called media. Media for plural, medium for singular. And that basically just gives the bacteria its nutrients to grow. Okay? And then we usually will incubate it in an incubator to keep it at a certain temperature that it likes to grow at. So there's two different types of media. There's solid media and there's liquid media. So liquid media is also known as broth. And it is just that. It is a broth. It is a liquid. So you put your bacteria into the broth, and they use that broth to grow, and they grow within the broth. These are pretty much always found in test tubes, but you can also put broth in larger bottles depending on what you're using it for. Then there is solid media. Solid media is also known as agar. Now, agar is the actual solidifying agent that's in the media. It is from a seaweed. It's an extract from a seaweed. And it is basically like the gelatin in Jell-O. So when you make Jell-O, you boil the water, you add the gelatin or the Jell-O packets, and you dissolve the gelatin, and then it hardens either in the fridge or at room temperature. Agar we treat it the same way. We have to boil the media to dissolve the agar, and then we autoclave it. Autoclaving is a sterilization process, and then we take it out of the autoclave and we let it cool at room temperature. And then gives us this hard surface to put the bacteria on. Now, there's a few different ways that you can see solid media because it is solid, so we're able to do more things with it. The most common way that you see solid media is in a Petri dish. Okay? And so that's the one when a lot of times when people think of media or agar, that's how they think of it being in the Petri dish. Okay? And there is two parts to a Petri dish. So I've got a Petri dish here. Okay? There is a bottom, and there is a top. The bottom is where the media is at. And I actually have media in here. Okay? And so that yellowy stuff is the media. And so I boiled it, autoclaved it, let it cool, and then I poured it into this Petri dish while it was still liquid. And then I let it harden, and so it's basically has the consistency of a hard Jell-O. And so that's a Petri dish. And when we are dealing with Petri dishes, we always want to store them inverted, upside down. That helps to prevent airflow and helps prevent contamination. Of course, in microbiology, contamination is what you don't want. That is bacteria that you are not interested in or fungi that gets onto your plate or onto your media and contaminates it. And a lot of times if you're dealing with an unknown something that you don't know what it is, then you don't even know that it's been contaminated. So that is a huge problem. So you can also see solid media in the form of a stab. And a stab is where we put the agar into a test tube, and when we let it solidify, we just leave it flat. So you have this surface, it looks like broth, but then you turn it upside down and it doesn't move. So we inoculate those with a needle and we stab down into the medium, and that's why it's called a stab. It's also called a deep sometimes, so if you search it, you might see that, a deep or a stab. And then lastly, we can see it in slant form. And in slant form, it's in a test tube, and after it comes out of the autoclave, before it cools, we slant the tubes. So it gives us this nice surface to place bacteria on. And if you were actually when you're actually coming into the lab and doing things in the lab, a lot of times the bacteria are placed on slants. And so this is just an easier way for us to store the bacteria. We can store it on plates, but it's also easier to store it on slants. So you could see it that way also. Okay. So now we are going to go over the procedure or the exercise for this, for the microorganisms in the environment. And basically, we're going to be using two different kinds of media. We're going to be using a general purpose media and then a more selective type of media. The general purpose media that we're going to be using is called PCA, plate count agar. And this is just a general nutrient agar that pretty much anything can grow on. And we are going to use that agar to basically show you where all bacteria are growing at and how easy it is for you to contaminate your sample by just touching something on your workbench or around you or anything like that. So what we're going to do is we're going to take our PCA, we're going to choose two different places to swab, okay, and then we're going to swab our media, and then we're going to look and see what all grew, okay. So that's what we're going to do with the PCA. Now, the PDA plate, that is the more selective media, and when we say selective media, usually it has something in the media that's going to inhibit the growth of things that we don't want. So PDA stands for potato dextrose agar, and this is selecting for fungus. So only fungi are going to grow on the PDA plate. And so with the PDA plate, we're just going to, I left a PDA plate out open to the air, and let the air in our lab circulate and touch the top of the plate, and then we are going to look and see what kind of growth we have on our plate. So let's go over to the workbench and do those procedures, and then we'll read the results. All right, so we are over here at the workbench to inoculate our PCA plate, and then I'm going to show you the results of the PDA and the PCA plate. So here's what my PCA plate looks like, okay, and it's just like a yellowy looking color. And so what we're going to do is we're going to choose two places to swab. Now, it can be anywhere in the lab or out in the hallway on yourself, on any of your stuff that you have, anywhere that you would like to see if there's any bacteria growing at, which there probably is. So the first thing that you want to do is you want to label your Petri dish. Now, you always want to label the bottom of your Petri dish because that's where the bacteria is. That's where they're growing. They're growing on the media, and the media is in the bottom of the Petri dish. So if you somehow get these two separated, the top from the bottom, you still know what's in this plate. Okay? If you only label the top, if they get separated, you have no idea what is in this plate because a lot of times you're going to have a whole stack of plates. So we always want to label the bottom. So I'm going to draw a line straight down the center, splitting my plate, not a very straight line, splitting my plate up into two different sides because I'm going to swab two different places, so I want two different sides. It's just going to help guide me, okay? And then I am going to label what parts that what I decide to swab. So I'm going to do the bottom of my shoe, and then I'm going to do the water fountain. And then I'm going to put my initials. You always want to put your initials on it, okay, so that you know that it is yours. Especially in here when we have lots of people that are going to be using the same shelf to store their stuff. Hey, so I've got 2 sterile cotton swabs. Now, normally the way I have these laid on the table, you would not want to do that because you want to keep them sterile. But this is just a demonstration, so it doesn't really matter. So 2 cotton swabs because we're swabbing 2 different places. And so basically all you're going to do is you're going to go and swab the area and then streak it back and forth on the media, on the plate. Okay? Now remember, the media is like hard jello, so you don't want to stab. Okay, the bacteria are going to grow on top of the agar, on top of the media. Okay, now advice for choosing where to swab. The wetter the surface, the more bacteria you're probably going to find. Because remember, these bacteria have to be living for us to put them on the plate and for them to grow on the plate. If you pick a very dry surface, a lot of times you're not going to get any viable bacteria. Bacteria need water just like we do. So if you pick a very dry surface, you're probably not going to get a whole lot. The bottom of your shoe usually always gives really good results because you've been walking around outside. And then the water fountain, which is what I've also chosen, gives really good results. Now, door handles and things like that, places in the bathroom, a lot of times they will give you good results because people are touching on those all the time. And then they have oil on their hands, so it's transferring and giving moisture. So usually the wetter the surface, the better your results are going to be. And so basically if I went and swabbed a place, then I would simply take my plate and swab back and forth on the plate. Now you want to swab a good bit because of course we want to see bacterial growth. So you want to swab really good. And then of course when I'm done with my swabs, they go in the biohazard because we don't know where we swab. We don't know, well, we know where we swab, but we don't know what kind of bacteria are there. So we want to make sure that these get thrown into the biohazard when you're finished. Okay, so I swabbed a plate and I put it in the incubator for 24 hours for it to grow up, and these are the results that I got from my PCA plate. So I did the bottom of my shoe and then I did the water fountain. Pretty much everyone's bottom of their shoes, unless they work in a hospital, are going to give about the same results, okay, because we're all walking in the same areas. So that's the bottom of my shoe. So you can see there's lots of different kinds of bacteria. So each one of those is called a colony. So that's where the bacterial cell landed on the plate, and then it has multiplied over and over again to give rise to the colony that you're actually seeing. So I want to be clear. What you're looking at on this plate is not an individual bacterial cell. It is a colony. So make sure that you don't get that confused. And next week, or the next video that you see, is going to be a simple stain. That's where we take a sample of a colony, and then we do a stain with it so that we can visualize the bacteria under the microscope. And those are the individual cells that you are seeing. The bottom is the water fountain. Now, you see that my shoe, because I'm walking outside, there's lots of different kinds of bacteria. They come in all different colors, the colonies do. Some are perfectly round, some have some weird edges to them. We've got some yellow ones, some orange ones, some white ones, some more colorless looking ones. So each one of those is a different kind of bacteria. Now you see the water fountain, those colonies are relatively small, mainly colorless because they're about the same color as the media, but they're all the same, right? So it's interesting because what's coming out of the water fountain is pretty much all the same bacteria, but then what's coming on the bottom of my shoe, it's got all kinds of different kinds of bacteria, okay? And so that's what we want to see for our PCA plate. Now, the PDA plate, remember, that's the one that selects for fungi. So only fungi are going to be able to grow on the PDA plate. And all I did with this plate is just open it. I didn't touch it. I didn't breathe on it. I didn't swab it with anything. All I did was open it up and let the air that's around in the lab touch the surface of the plate. I did leave it out overnight. Now, if we were in the lab, we would only leave them out for about 30 minutes or so. But I left it out overnight just so I would get some really good growth. So, you'll see that the fungus looks completely different compared to the bacterial colonies. They look hairy, okay, or fuzzy. That's the word that I usually get in the lab if we were actually looking at these in the lab, okay? That's called filamentous growth. So fungi colonies give off these hair-like projections, and those are filamentous. So that's going to be the main difference between being able to tell the difference between a fungal colony and a bacterial colony. If we look back at our bacterial colonies, you'll see that they're not really that raised, and there's not those hairy, fuzzy projections coming up off of them. So if you were in the lab and you had an unknown that you were working with, and say all the bacterial colonies looked like these that were on the water fountain, and then all of a sudden you got something like this that was growing on your plate, you then clearly know that that is not your bacteria. You've got some kind of contamination, and you need to address that. You need to do another streak plate, try to get it pure again. So fungal colonies come in all kind of shapes and sizes and colors. Sometimes you'll see black. These are kind of a green color. You'll see that there's an orangish looking one. Okay, the inner is orange. So lots of cool colors. They form these indentions into the media, so they grow into the media. So these are going to be your fungal colonies on PDA. Okay. And so again, the point of this exercise is just to show you how easy it is to contaminate your culture in microbiology. We want you to practice aseptic technique so that you're not contaminating things, whether you're working in a microbiology lab one day or a hospital one day. We want to make sure that you understand that bacteria are everywhere, fungi are everywhere, and we don't want to contaminate our samples.